Okay, so what we were discussing in the last class was we were talking something about endomembrane system. So what we remember from the discussion that we had in the last class, endomembrane system boli kole, what we understand? What is endomembrane system? Or who comprise the endomembrane system? Certain organelles, right? What we have said that inside the cell there are certain organelles, and some of the organelles they always work in a coordinated manner, and ultimately they form the endomembrane system. Talking about those organelles that belong to the endomembrane system, ki asile? Endoplasmic reticulum, Golgi bodies, lysosomes and vacuoles, right? We have started with the endoplasmic reticulum, and today we are going to continue with that itself, because in the last class we have not completed the endoplasmic reticulum. So we will be continuing with that. Before starting or to write the notes, what we remember about endoplasmic reticulum? Ki monot ase? Position to kunkinit. Where will you find? Attached to the nuclear membrane, right? Next, talking about the parts of endoplasmic reticulum, it is made up of how many parts? Three parts. It is cisternae, then vesicles, aru tubules, right? Cisternae ki asile sac-like structures, and if you talk about vesicles, these were certain globular or circular structures, and talking about tubules, tube-like structures, right? Next, types of ER, two types to be precise, that is RER and SER. The differentiation is on the basis of presence or absence of ribosomes. If ribosomes are present, then RER, if it's absent, then it's SER. Talking about the most important functions of RER and SER, apart from the various points that we have written, so what's the important point? Synthesis of dash and synthesis of dash, synthesis of proteins and synthesis of lipids and sterols, right? Talking about synthesis of proteins, kune synthesize korisile? RER. Are talking about lipids and proteins, lipids and sterols, it was SER, right? So junkhini stop korisa, tarpore continue korim. First likha points boli likhasun. Last time ki likhisu? A comparison between SER and RER, right? Okay, so this is a point that has a relation from the point that we have discussed in the last class. So if a question is asked like this, primary lysosome is formed by, or who forms primary lysosome? Answer ki hobo? Who forms it? Could I say answer to correctly? It is RER, because that was the last point you have written in the chart, just see, right? But now we know that primary right, because after this we are going to study Golgi bodies, and Golgi bodies are also responsible for the production of primary lysosomes. Talking about lipid synthesis, SER, aru eta structure. Remember this word? Just look into the chart that we have discussed. That's why plant lysosomes are called as ki? Peroxisome, right? So ultimately, lipid synthesis kune koribo both SER and peroxisomes. So it is a point monot rakhibo laibo. After these 2 points, likhiba functions of ER. Functions of ER boli likha. Etur report kenekwa questions ahise we will be discussing. First write this. So functions of ER আকৌ এখন chart বনোৱা এটা side SER on the other side write RER, just make it in the form of a chart. Detoxification of drugs, DRUGs. In the last class, I think we have said something about peroxisome, no? কি কৈছিলো? The inner cell, it's not necessary that we will see only the bigger cell or cell organelles, but apart from that, কিছুমান smaller organelles থাকে, যেনেকে one example was perosomes, the other example is peroxisome, right? For those students who have studied something called as a C2 cycle, they might be knowing about peroxisome. C2 cycle ক'ত পঢ়িছিলা? First year to পঢ়োৱা নাই? C2 cycle ক'ত পঢ়িছা? C3, C4, C2, CAM cycle, এইখিনি ক'ত শুনিছিলা? Photosynthesis. Right? Photosynthesis kuntu part of light reaction, dark reaction. Dark reaction, right? So if you have studied about the C2 cycle, we know that it is one cell organelle where the C2 cycle also takes place. C2 cycle boli kole one point that we remember, it is also called as 3-cell organelle process. So 3 cells are involved, right? Next, one basic difference between C3 and C4. C2 cycle is present in one, absent in one. I probably piece out point C3 plants, C4 plants. Okay, as of now, detoxification peroxisome of liver cells. Kuntu provides eto precursor, PRECURSOR, precursor mane raw material. Simple meaning is raw material. Anything, if you have to produce something, tar agote amak ki lagibo? Raw materials lagibo, right? So if enzymes have to be formed, then something that forms the enzyme, that is what is called as precursor, right? So who helps in the formation of lysosomes in the Golgi bodies? It is the RER. So some of the functions of ER or some precise functions of SER and RER. Maximum question has been asked about. Obviously point number 1 to question ahe lipid sterols kune produce kore, proteins kune produce kore. Next is detoxification of drugs. Etu upor to question ahe, muscle contraction upor to question ahe. Just the words are important from MCQ point of view. Details has not been asked. ठीक है so who detoxification code hoy obviously in the liver cells. Who helps it? It is the SER, right? So junbilak the people who have a high, I mean who consume a high range of or huge amount of alcohol, tetiyahole ki hoy? I think you all have heard, no? Alcohol consumption or ki hoy? Up to a certain extent the liver remains quite good, but after some time ki hoy? Liver to deteriorate koribo arambho kore, no? What is the reason behind? Because detoxification aru no hoy. Right, there is a limit of detoxification in the liver cells. So if alcohol is taken, it gets mixed with water and ultimately the waste product is passed through the urine. This is the function that is done by the SER that is present in the liver cells. You know, they also have a limitation of working, right? After a certain period of time, the amount of alcohol is much more, the liver starts degrading, right? So iman details nalage khali 8 point more. Muscle contraction or logot kun associated SER. I think another point that you remember about muscle contraction is if you have read the chapter called as respiration, aerobic respiration, anaerobic respiration. So anaerobic respiration, muscle contraction not muscle contraction janu ni najan. Sometimes ki hoy muscle nerves ba nerve cells bilak ki hoy tani dhore no. So that is also a reason because of what? Because of anaerobic condition that occurs inside the cell. Right, so muscle contraction, something that we are going to study here and also in the chapter called as respiration. As of now, just the words are sufficient, right? So these are certain functions of SER and RER. Next, likha general functions of ER. General functions of ER. So not very important, just certain general points. Coming to the last point, that is modification of protein. Generally the word modification of protein is associated with Golgi bodies, but we see that certain types of modification also occurs in ER. But from examination point of view, if the question is asked modification of proteins occurs in, it's always Golgi body that you will write, right? This is an added function of this one, so I have written this word mainly, right? Eta dutaman question likhim if this is over. Was this over? questions pure likhi number 1 likha the cell organelle just to have an idea say ekhini portion opor kenuka question ahise the reason for giving such a question is just to see the difference, right? You have to answer according to the option that is given in your question, right? Eketa question not ever ekhini options asile ever ekhini options asile. So if this is the first question, just don't think about this one now. If this is the question, then everyone knows that we are talking about what? SER, but sometimes a question might come this way also. We know lipid buli kole khodai ki monot thake SER kintu option to SER nai. Does that mean the question is wrong? No. Right? In that case, ki hobo? Spherosome. Right? So this is just a way to tell you that please read the question properly and answer according to the option that is given. You don't have to fix it in your brain say lipids boli kole khodai SER ei hobo lagibo tenuka nohoy. Right? Option wise answer to koribo lagibo. Akko eta question likha, question number 2. Is this part over? Etu okay, hetu likhi lua. This one, amyloplast, AMYLOPLAST. Amyloplast is nothing but a plastid. Again the same thing, right? One question having different options. We know whenever we are talking about primary lysosomes, yate question or answer ki hobo? This one, but in this case only Golgi bodies, right? So I think now you have an idea. So depending on the options, you have to correct the, I mean you have to choose the answer, right? Next we have to write a single point about RER. Next okoman heading koriba RER. So remember when we were discussing endoplasmic reticulum, once I have asked the question saying that in a cell protein synthesis kot hobo. Where will protein synthesis takes place in a cell? Wherever ribosome is present, protein synthesis will occur there. Now where will you find the ribosome? This answer is regarding that drawing. If you see, this is a rough diagram of a cell, right? So if you are talking about the cell, you will see the presence of ribosome in the cytoplasm and also the presence of ribosome in the cell organelles also. So abelic ribosomes also they will produce proteins, these ribosomes will also produce proteins. Now you have to know what will happen to the proteins that is synthesized, right? So they will also synthesize protein, they will also synthesize protein. What happens to the protein that is formed? Some proteins or some type of proteins remain within the cell and some proteins goes out through the plasma membrane, or it gets occupied, or it gets it is used by the plasma membrane. Kisuman plasma membrane use kore, kisuman eta cell or antu cell le gusi jay, aru kisuman ki hoy within the cell it remains confined, right? So a diagram to likha and then we will write the points. Below the diagram, write ribosomes of endoplasmic reticulum. What they do? They make proteins that remain in the cell, or the proteins may go outside the cell, right? But if you are talking about ribosomes in the cytoplasm, they will make the proteins and these proteins will always remain inside the cell, right? So as far as this diagram is concerned, so if you are talking about these type of ribosomes that are in the cytoplasm or inside the cell, tahate jinbilak protein synthesize koribo, these proteins will always remain within the cell and they will never go out. But if you are talking about ribosomes that is inside or on the endoplasmic reticulum, tahate jinbilak protein synthesize koribo, they might stay in the cell or they might go out to the next cell also, right? So this is the basic difference. So this was all about endoplasmic reticulum. We have done enough regarding endoplasmic reticulum. So the first organelle of the endomembrane system. So we'll be now moving to the next one, that is Golgi body, heading koriba Golgi body, if this is over. Is this over? Buji to paisu, no? Nothing very hard or tough, right? But you need to study, then only you will remember. Bahut kini contents likhisu, no? The note is quite long, so please keep on reading so that you remember. Okay. Talking about Golgi bodies, no? Golgi body boli kwa loge lage. What is the most important function that we remember? What is the function of Golgi bodies? Ki kore? What is the most important function of endoplasmic reticulum? Make proteins. What is the function of Golgi bodies? Modifies proteins, packing proteins, right? After packaging, what happens to the proteins? It goes away or it goes to the respective targeted areas, right? Jote lage tale gusi jai. So whenever we talk about Golgi bodies, that's the first line we remember. Next, Golgi bodies are found in all the cells, almost all cells, but the number of Golgi bodies is more in a certain type of a cell. Kuntu cellot sobotse beshi Golgi body thakibo. The cells that will secrete certain things. For example, if you are talking about the root tip or the root cap, right? So root cells or Golgi bodies maximum thakibo kiho karone they will secrete a mucilaginous substance that will help the root tip penetrate towards the soil. So Golgi bodies thakile ki hoy secretion of mucilage hoy. Secretion of mucilage maximum ami kot dekha paisu in the root cells. And it has to penetrate. As the soil particles are obviously they will have to have certain type of lubricating agent, right? So kune produce kore? Golgi bodies produce kore. So okay, Golgi bodies aapko let's write the note first. বহুত কিটা নাম আছে বহুত নাম এই আগৰ দিনৰ মহাৰাজাবিলাকৰ বহুত নাম থাকে ন something like that. Finally, it's over. I think I have spoken about dictyosome previously, right? In plants, Golgi body present or absent, obviously it's present, but it is not given the term Golgi body, instead it is said to be as a dictyosome. What is the basic difference between a Golgi body and a dictyosome? In case of a Golgi body, tahator ki hoy, the cisternae are very compactly packed, right? So if you are talking about this flat structures, these are very compactly packed, and if it's compactly packed, the structure is called as Golgi body. But if the cisternae are far apart, right, tetiale tahatok ami ki boli kom? Dictyosomes. Golgi bodies present in, absent in mature sieve tubes, mammalian RBC, sperm cells of bryophytes and pteridophytes. Sperm cells mane the male gametes, right? Mature, mature. Talking about the structure, key shape thake, it is unbelievable. It is a C-shaped structure. So, Golgi body is C-shaped structure, concave or convex side we understand, right? Convex side to that a particular name that is called as the cis phase, and the concave side is called as a trans phase. Another point is maturing side of the Golgi body is called as the trans phase. We will be writing about it. So, the phase towards the plasma membrane is called as trans, the phase towards the nuclear membrane is called as cis. You have to read the terms, the names properly, right? Which is the cis phase, which is the trans phase, question ahead report, right? The phase towards the nuclear membrane, the phase towards the plasma membrane. ইমান প্ৰপাৰ একো আঁকিব নালাগে So talking about the structure of Golgi body, we know that it's a C-shaped structure. It has two faces, the cis and the trans. It has a concave face and a convex face, right? So if you are talking about the concave face, then that is called as what? The trans phase, which is also known as the maturing phase. Why is it called as the maturing phase? Because it is through this phase that the packaged proteins moves out from here. Right. So forming phase in the sense, when we are talking about a Golgi body, it is something that belongs to the endomembrane system, so obviously it does not work independently. So it is connected with the endoplasmic reticulum. Exactly what happens is the proteins that are synthesized in the endoplasmic reticulum from the ER goes to the Golgi bodies, and when it goes to the Golgi bodies, obviously it will be entering through the cis phase, and so this phase is called as the forming phase, right? Iate junbilak vesicles ba junbilak vesicles form korise, a vesicles bilak uporle ba maturing phase of goi like ki hoise coated ba packaged hoy goise. Once it is ready, it goes away from the cell. So that side is called as the maturing phase. Talking about the parts of a Golgi body, as we have already discussed, no, it's something in common with the endoplasmic reticulum. Endoplasmic reticulum hoto ami keta part paisilu, tinta. Cisternae, tubule and vesicles. In case of Golgi bodies also, we see cisternae, tubules and vesicles. Apart from that, one special structure that is called as the Golgi and Vacuole, right? So the question that has been asked about Golgi and Vacuole is, Golgi and Vacuole in the Golgi body is formed in dash phase. It is formed in the cis phase, it is formed in the trans phase. So it is formed in the trans phase. What is the function of the Golgi vacuole? It is that vacuole or the vesicle that will ultimately work as a lysosomes. So lysosomes kune produce kore, Golgi bodies produce kore, right? So lysosomes boli koa loge loge, what we said is lysosomes will be studying about it, and we know that there are 3 types of lysosomes: primary, secondary and tertiary lysosomes, right? Primary lysosomes mane ki the first form lysosome. Who forms that lysosome? As we have discussed, it is also the endoplasmic reticulum and also the Golgi bodies, but mainly hey vesicle too. So lysosome boli kole ki hoy, it is nothing like a sac like structure. Inside the sac, what you see? Certain types of enzymes inside it, right? Acid hydrolase by acidic enzymes kisuman ase, right? So ultimately lysosome to form koribole jinbilak enzymes lage kune form koribo, endoplasmic reticulum form koribo, structure to kune form koribo, Golgi bodies. So that is the reason why we give the credit to both the cell organelles, right? But basically if the question is asked who forms Golgi bodies, and in the option, sorry, who forms lysosomes বা primary lysosomes, our option of যদি অকল Golgi body আছে, answer would be only Golgi body. Clear, no? Okay. So this is important, etu saba. Next we are going to write something about the 3 parts: cisternae, vesicles and tubules. Is the diagram over? The heading koriba cisternae. Online students, is the diagram over? Okay. Okay. Cisternae boli heading kori. What is a cisternae? It is a flattened sac-like structure. So what is a cisternae? It is a sac-like structure. They are connected with each other, as we have already discussed. They almost resemble SER because they do not have the ribosomes. So they look like SER. They are arranged near the nucleus. No doubt we have written this line, but I think we know the placement of the Golgi bodies. If you are talking about the cell, if this is the nucleus, next to the nucleus, is it the Golgi body or it's the endoplasmic reticulum that you are going to see? It is the ER, no? ER khini ami dekha pam, tar pisothe ami ki dekha pam, the Golgi bodies dekha pam. So obviously it's near to the nucleus, and that's the reason we have written that statement. That does not mean the cell organelle next to the nucleus is Golgi body, hetu nohoy kintu. Next to the nucleus ki hoy? Endoplasmic reticulum hoy, right? (0.5 micromu to 1.0 micromu. Nothing to write, arises from the periphery of cisternae. পেরিফেরি বুঝি পাও ন সাইড Why they are called as traffic point? Because they do the work of carrying substances from one part to the other part. ট্ৰেফিক পইণ্ট বুলি কিহৰ কাৰণে কয় কাৰণ তাহাঁতে কি কৰে ভেচিকলছবিলাকে প্ৰটিনছবিলাকক এটা পাৰ্টৰ পৰা আনটো পাৰ্টলৈকে কেৰি কৰে এতিয়া ফৰ এক্সাম্পল এণ্ডোপ্লাজমিক ৰেটিকুলামত যোনখিনি প্ৰটিন চিন্থেচাইজ কৰিলে ন সেই প্ৰটিনছখিনি ক'লৈ যাব লাগিব গলগি বডিজলৈ যাব লাগিব চ' হাউ উইল দে গ' দে উইল বি কেৰিড এলং বাই দ্য ভেচিকলছ right so sac like structures I'll be drawing a diagram after we complete this part and then it will become more easy বুজি পাইছানে ট্ৰেফিক পইণ্ট কিয় কয় এজ অফ নাও বিকজ দে আৰ ৰেছপনচিবল ইন কেৰিং দ্য ছাবষ্টেঞ্চেছ ৰাইট চ' দিছ ইজ অল এবাউট দ্য ষ্ট্ৰাকচাৰ নেক্সট লিখিবা ফাংচনছ অফ গলগি বডি two important words from MCQ point of view, not in details, glycosylation and glycosidation, right? Details ami beleg eta chapter dia ase. Glycosylation of proteins means formation of glycoproteins, glycosidation is formation of glycolipids, right? So yata question ahe, the organelle associated with glycosylation and glycosidation, right? The organelle associated with glycosylation and glycosidation of lipids and proteins is nothing but Golgi bodies, right? Only 8 term or question ahe. As far as formation of glycoproteins, formation of glycolipids, what we understand, the protein to already produce korile, kune produce korile, endoplasmic reticulum, right? This proteins comes in combination with some other particles and ultimately they will form a compound. If the protein comes and joins together with what? Glucose, then they will form what? Glycoprotein, right? Jedi lipids ahi pelai glucose logot log lage, glycolipid. So beleg bostur logot log lagibo pare. Proteins originally code form hoise endoplasmic reticulum. Yate ki hoise? They come and combine with a different other compound. I mean other substances and ultimately they form different compounds, right? So 8 word one rakhiba. Then number 3. Yate aru eta word likhasun. The first point. Because two main words, modification of proteins and packaging of proteins. Right? If this is over, ako kisman points likhim, points puli heading koriba. Functions of Golgi bodies opra hodai question ahe. So first, second, third, specially, right? Recycling of plasma membrane means we are talking about reformation of plasma membrane. Sometimes what happens is plasma membrane might get destroyed, it might get broken down also. So ako form koribo lagibo na lage jedi plasma membrane kon break down hoise he kon to ako form koribo lagibo. So form koribo lagile obviously it will require certain types of proteins, and who is going to give it is obviously the Golgi bodies, right? So that is what we understand by recycling of plasma membrane. Okay, next points, sort ketaman points. Is this over? So while we are writing a note, obviously if we write a note in sentences with proper grammar, etc., etc., the note look like it seems to be quite long. But if you, I mean, as I said, no, if you have the habit of making a short note, then iman dangor notes ketiao nohoy. It is just words that you will pick up and you write in your short note. Golgi bodies are protect to sentence you don't have to remember, right? It is those points that is important that you write it down apart, right? Tetia hale ki hobo study bostu to komi jabo. Golgi apparatus or Golgi bodies, ye kwa? Eukaryotic cells of Golgi bodies thakibo, fungi eukaryotic ne prokaryotic? Obviously eukaryotic, right? But in spite of that, you will not see a proper or well-defined structure. Instead, you will see a Golgi body that is unicisternal. Unicisternal mane comprised of just a single cisternae. Eta thakibo matro. Legs, okay, legs are well-formed Golgi body. Okay, a taller line to dekha paisa. Okay. Now, from examination point of view, ki monot rakhim. I think looking at this, you have an idea what we understand by the word modifies or packaging, right? So modifies, what it modifies proteins, it modifies lipids also, right? So lysosomes are the vesicles that have a particular type of enzymes. What is the enzyme that we are talking about? Hydrolytic. Hydrolytic, what we understand by the meaning? Water or lytic, breaking down, right? So bigger substances into smaller substance, breakdown kune kore lysosomes or bhitor thaka enzymes kiniye, right? So these vesicles, if they contain hydrolytic enzymes, those vesicles are called as lysosomes. If they do not contain hydrolytic enzymes, they are just called as vesicles and not lysosomes. Clear? So this was all about Golgi bodies. Eta point about ER ami miss korisu. Jedi endoplasmic reticulum or such jagah ase tate likha, or else you can write it as an extra point also, doesn't matter, right? But if you hear about SER, these are the things that you have to remember, and not only lipid synthesis. Take the lipids or sterols to obviously produce koribo, that's something that's very common. Detoxification is a word, and obviously calcium storage. Remember, we have talked about muscle contraction, right? That point out my calcium mention korisu. So question ahe calcium storage logot kun related, it is a SER. Right. So endoplasmic about endomembrane system we have completed 2 structures. Next likhiba lysosomes. Lysosomes likhimle vacuoles likha. Third likhiba vacuoles. Is this all good? So talking about a vacuole, I think we understand what a vacuole is. What is a vacuole? It is an organelle. How will you define a vacuole? Vacuole keneke define koriba? Sac like structure, round structure, all round and different shapes hobo pare. Talking about vacuole, another point that we know is it's a major characteristic or main characteristic of a plant cell, right? So talking about the location of the vacuole in a plant cell, it is central, quite large, central, the largest organelle in a plant cell, etc., etc. Then we have to talk about types of vacuoles, right? It is not only a vacuole that is present in the plant cell, that vacuole also has a specific name to that, no? Sap vacuoles, contractile vacuoles, sap vacuole, gas vacuoles. So there are certain types of vacuoles. Next, as far as the definition of a vacuole is concerned, I mean vacuole keneke define koru? It could be said as a non-cytoplasmic area inside the cell. Is it correct? Non-cytoplasmic area inside the cell is called as vacuole. Is the statement correct? Cytoplasm ki asile? the liquid or the gel-like substance that contains all the cell organelles. Inside the organelles, cytoplasm থাকে নেকি? নাথাকে। তো ভেকিউলৰ ভিতৰতো কিবা এটা সাবষ্টেন্স থাকিব ন, সেইখিনিক আমি সাইটোপ্লাজম কম নেকি? নকওঁ, right? So that inside content, especially in case of a plant cell, is called as cell sap, right? So if you are talking about a cell like this, especially plant cells যদি কথা কোৱা, right? So you have this area, right? এই বাহিৰখিনিক আমি কি বুলি ক'ম? Cytoplasm. এইটো যদি vacuole হয়, so this is obviously not the cytoplasm. So what is a vacuole? The non-cytoplasmic area inside a cell is called as a vacuole. Clear? And talking about a plant cell, আৰু এটা term কি জানিব লাগিব? Something called as a tonoplast. The boundary of a vacuole is called as a tonoplast. Next point about specially, so out of the all types of vacuole, maximum discussion has to be made on plant cell যোনটো vacuole থাকে ন, that সেই vacuole details লাগে, বাকিখিনি vacuoles আমাৰ ইমান details নাথাকে. So next point that you need to know is what is there inside a vacuole. Vacuole এটাৰ ভিতৰত কি থাকে? What is the function of a vacuole? Storage. What does it store? Many type of materials, and together with that certain what? Waste materials also. Very good. Some certain types of waste materials also. Next, one important storing compound is something called as pigments. Kisuman pigments u thake, right? So I think you all have heard something called as water soluble pigments or lipid soluble pigments. শুনিছানে? Water soluble pigments, lipid soluble pigments. Especially lipid soluble pigments বিলাক কোনবিলাক? The pigments that are associated with what? Photosynthesis, chloroplast, lipid soluble ne? Water soluble, lipid soluble. But if you say of something called as anthocyanin, water soluble, right? সেইবিলাক আমি এটা discuss কৰিম. Pigments তাৰমানে কি হয়? Broadly classified into two categories, water soluble and fat soluble or lipid soluble. Water soluble pigments বিলাক ক'ত থাকে? Vacuole এটাৰ ভিতৰত থাকে। কিন্তু lipid soluble pigments ক'ত থাকিব? If I say chlorophyll is one example of a lipid soluble pigment, ক'ত থাকিব? Inside the chloroplast. What is a chloroplast? Nothing but a plastid. So pigments দুভাগত ভাগ কৰিছো, water soluble, fat soluble. Water soluble ক'ত থাকে? Vacuoleৰ ভিতৰত. Fat soluble ক'ত থাকে? Plast বা chloroplast বা plastidৰ ভিতৰত. Clear? So vacuoles are first statement likha, non-cytoplasmic area inside the cell. Inside the cytoplasm বুলি লিখিলেও হ'ব. This would be more precise. So out of this 4 details অকল sep vacuole লাগে বাকি কিটা ইমান ইম্পৰ্টেন্ট নহয় Osmotic pressure, শুনিছা আগতে, right? What is osmotic pressure? The pressure that is exerted on the osmotic pressure is a pressure. Where is the pressure exerted upon? Osmotic pressure is something related with osmosis. What is osmosis? Something related with liquid, right? What happens in osmosis? High concentration to low concentration, or you can say what? Low concentration to high concentration. Concentration of what? Concentration of solutes, or either you are talking or you are saying the definition in regards to high concentration and low concentration of water molecules. Jodi solute ami consider koro, then we know that we are talking about the movement from low to high. Low solutes to high solutes. The water will always move, right? If you are talking about one cell here, one cell here, it has 3 solutes, it has 5 solutes. Pani kopra kole jabo, right? What you have to remember, the cell will drink water only when it is eating many solutes. Bhat khale ki hoy? Pani khao. Solid bostu khale ki koru? Pani khao. So cell to solid bostu kot besi ase? Yate besi ase. So pani kune lobo? Jot solid besi thake tate pani tu jabo. That is one line that you remember, and this is the only statement that you have to remember while reading the chapter called as transport in plants. The entire chapter revolves around this only. যে movement of water kuntu cellor pra kuntu cell le jai. The water always moves from a cell, or it moves to a cell that has more solutes, right? Solute jedi besi pani tale jabo, right? That is one concept. Okay, talking about osmotic pressure. Osmotic pressure, osmosis we understand, it is the pressure which allows the movement, right? Pani yar pra yale jabo across a semi permeable membrane. That's a word that you have to use while defining osmotic pressure, osmosis. Now talking about osmotic pressure. Osmotic pressure ki asile? It is the pressure that is exerted on ki hot solution not ne solvent on. Karon osmosis boli kole dutai to concern, no? Either we are talking about the solvent or we are talking about the solution, because this water will move from where to where? Solvent to solution, solution to solvent, because we are concerned only with the liquid part, right? So since we are defining osmosis with the two words solvent and solution, the pressure to kot exert koribo. Osmotic pressure hobo lagile, pressure to kot exert hobo. It's the pressure on the solution or it's the pressure on the solvent? It is the pressure on the solution to stop the entry of solutes into it. It is exactly like reverse osmosis. Osmotic pressure ki hoy? As of now, since we have not done that chapter, no? So osmotic pressure is nothing but reverse osmosis. Osmosis we know, just the opposite of osmosis ki hoy? Reverse osmosis, kiba eta process opposite hobo lagile, kiba eta extra koribo lagibo nalage. Osmosis is something that will go on naturally. If you want to reverse the process, then obviously you have to do something. And what is that something we are talking about? You have to exert a pressure from outside. So while exerting a pressure, quote pressure to dim. Solution or the solvent out? It's always the solution. Just remember RO. I think you have heard that word called as RO. RO is associated with what? Water purification. What is water purification? Ki kora water purification out? You have dirty water, you remove the water molecules and separate the solute and the liquid, right? Hey, say, so, eta chamber ase, etu eta chamber ase, thik ase? Yate ki ase? Solution ase, solute water. Yate ki ase? Okol water ase, right? Yate ki ase? Solution. Normally solutes ko besi ase, yate. So normal osmosis at ki hobo? Etu chamber pra etu chamber le ahibo. Reverse osmosis at ki hobo? Etu pra etu le ahibo le. So if you are talking about filtration, you want to filter this liquid, this water that is dirty. What you will do? You will take this liquid, put it to this. That is what is called as RO, right? So yar pra water molecules bilak jodi yale jabo lage. Obviously through the semi-permeable membrane, since we are talking about osmosis, tetale yate ami ki dibo laibo? Eta extra pressure dibo laibo, right? A pressure to ke ki boli koy, right? Osmotic pressure, we will define it later. As of now, what is osmotic pressure? It is a pressure on the solution, on the solution chamber to prevent the entry of solute into it. Ami pisot define kori, right? Okay. This is over, no? Sap vacuole. Sap vacuole aru eta point likha. Points about sap vacuole. A term. I think it's understood. You have a proper term for that amount of cytoplasm that is present in the cell. Especially with cell we are talking about, obviously a plant cell. Because cytoplasm content kini plant cell or kom thakibo kyo karone the center itself is occupied by a big vacuole. So if this is occupied by the vacuole, then this vacuole, and we know vacuoles bilake grow kore ne nokore. What do you know? Okay, do you know something about the growth of a cell? Does a cell grow? Cell bilak grow kore nokore? Cell living and non-living? Living. So grow koribo nokore? Keneke grow kore? Keneke kore? Who is responsible for the growth of cell? Cell division. If you say a cell grows by cell division, that is a statement that is related only with the unicellular organisms. A bacteria grows by cell division, but if you are talking about a human being, amar cells bilak bachate jiman dangor asile etio himanei dangor ne take dangor hoise? Obviously dangor hoise. To keneke dangor hoise? Cell division korise niki? Liver we had 5 cells in when we were babies, and now we have 10 cells, and little later we'll have 100 cells, and the liver will keep on growing like that. That is what we mean by cell growing? No. A cell grows because of the vacuole, right? Vacuole absorb koribo. Right? We'll be talking about it later. As of now, bad dia. Ekin loke ami buzi paisu. Primordial. Etu khali word to janibo laibo. Pigments kini ekini upor question ahe. Example of a pigment, anthocyanin we have already written, xanthin we have written. Anthocyanin boli kole another point that you need to remember, not in this chapter, beleg eta chapter, something called as a beetroot. Beets chini pao no, ronga beet juntu. Hey, beet to panit thoi dile ki hobo? What will happen if you put the beet in water or if you boil the beet? The water will become red. Why? Kiho karne? Because the pigment that was present there comes out in the water, right? So that is why it comes out with the water, or why it mixes with the water, because it is water soluble, right? Kine eta green leaves jodi panit thoi dia, pani khini green hoi jabo niki? Na thake, kiho karone? Karon green leaf to pigment thake, but that pigment is stored in what? Plastids, so it will not come out easily. But if it is stay in a vacuole, it's easily that will come out, right? I think this is all for today. We'll be continuing about vacuoles in the next class. Cytoplasm in a plant cell due to the presence of a large central vacuole. Plant cell to vacuole to bahut dangor hoi thake, he karone cytoplasm content to bahut mihike thake. That amount of cytoplasm has a particular term that is called as primordial article. Clear? Okay then, thank you.